Welcome to this video, where we will talk about INRA MetaGenoPolis, Center of Excellence for Human and Animal Microbiome Research. The MetaGenoPolis is pioneering the industry with more than 80 scientific publications in prestigious journals such as Nature, more than 33 patents, and 3 spin-offs such as Untitled Chapter, and Excellium. The main process is composed of 3 steps. First, the biobank, which is here to store human and animal samples. The sequencing part, which is there to extract the genome. And the data analytics part, to do statistical analysis, AI and machine learning modeling, biomodeling, etc. The biobank is currently storing more than 20,000 samples and has a total capacity of 600,000. It follows standards agreed with 30 institutions for collection and extraction to ensure quality of the information stored. INRA is currently sequencing more than 100 samples a week, with 7 ion protein sequencer and soon an Oxford nanopore minion sequencer. Now let's talk about the data analytics part, where we will talk about workload distribution, dynamic resource pool, multi-language workflows, and visualization. Now let's move to the resource manager, where we will see all the connected resources, in our case, one VM with 8 nodes. In the Workflow Studio, you can visualize the pipeline that is used during this demo. On top, the replicated task in Ruby that will be applied on all the samples, and at the bottom, some tasks in R to visualize the result in the browser. Let's execute the workflow. We can then move to the scheduler portal. This is where you can fetch the logs, visualize the progress at the tasks level, manage errors, and more. In our case, we mapped one processing task with one VM since it's multi-threaded. We've set up earlier a scaleset INRA node source, which is comprised of a dynamic policy and an Azure scale set. This means that when the scheduler load increases, the resource manager will automatically fetch new resources and connect them to the resource manager. Those resources will then be available to process tasks. We have now 4 VMs connected to the resource manager, processing 4 different tasks. Finally, when the load decreases and the resources are idle for a certain period of time, the resource manager will automatically release the resources and ensure that you don't pay extra. We now have one final sample to process, so we kept one VM alive. In the end, some final task will generate reports. In the scheduler, we can see the 5 samples processed. Let's now visualize the results. We can now see the statistical analysis made on all the 5 samples and understand better each patient. Finally, we have a dynamic and elastic resource pool which is fully utilized during the whole process. This is obviously a small part of what INRA can provide. You can see a full list going in front of your eyes, such as big data analysis, biomarker identification, and a lot more. Do not hesitate to contact us if you are interested in our work on the human body, on the animal. We tackle markets in pharma, agri-food, nutrition, clinical, animal nutrition, health, etc. Thank you for watching.